Okay. So, thank you. I will present my thesis concerning the development of a new therapeutic approach for Alzheimer's disease. So briefly, Alzheimer's disease is a neurodegenerative disorder characterized by a loss of cognitive functions, including those related to memory, reflection, language, and mood, and is linked to a synaptic dysfunction and a local neurodegeneration. Two main features are found in Alzheimer's disease: amyloid plaques composed of beta-amyloid peptides and neurofibrillary tangles of tau protein. Currently, no efficient treatment is available, and existing medications are only symptomatic in order to improve the life quality of patients. According to the WHO, more than 35 million people live with dementia worldwide, comprising 60 to 70% with Alzheimer's disease, and the absence of treatment involves an important economic cost estimated at $604 billion in 2010. The identification of new therapeutic approaches is this an important challenge for the future. Phospholipase A2 are involved in the cleavage of fatty acids of membrane phospholipids and thus in phospholipid metabolism, membrane turnover, and intracellular signaling. However, their roles in Alzheimer's disease were newly discovered. Indeed, some phospholipase A2 isoforms are overactivated in Alzheimer's disease, leading to inflammation and mitochondrial dysfunction, and thus in reactive oxygen species production and in apoptosis. Further, reactive oxygen species are able to stimulate kinases involved in phospholipase A2 activation. And beta-amyloid will accentuate this vicious circle by inducing excitotoxicity through an NMDA receptor and by its involvement in mitochondrial dysfunction. Finally, the development of new approaches concerning the central nervous system requires to be interested in the blood-brain barrier. It is an impermeable barrier between the blood and the central nervous system. The vascular unit is composed of endothelial cells of brain capillaries sealed by tight junctions and surrounded by pericytes and astrocytes. This barrier restricts the crossing for more than 98% of molecules, including new molecules developed for treatment or diagnosis of brain diseases. And thus, the development of new crossing strategies is a key step when the target is the central nervous system. Existing methods for the crossing of this barrier include invasive and non-invasive methods, and we use the receptor-mediated transcytosis for the development of the blood-brain barrier shuttling. And we selected the LDL receptor involved in cholesterol metabolism. Its endocytosis in the whole body is involved in the cholesterol degradation, but in the blood-brain barrier, this receptor is involved in a transcytosis. And thus, the LDL receptor is a receptor of choice for the blood-brain barrier shuttling. The objectives of this project is the development of a therapeutic strategy by targeting an isoform of phospholipase A2 and a shuttling strategy by targeting the LDL receptor. The targeting is performed by using the phage display technology allowing peptide identification. The phage display is a high-throughput screening method allowing peptide identification with affinity for a target, and is based on a round of selection of a peptide library where each phage expresses a different peptide. After target immobilization, the peptide library is incubated with the target, and phages with affinity will bind this target. After washing steps, the bound phages are eluted and amplified in Escherichia coli in order to obtain a new pool of phages that will enter in a new round of selection. During a round of selection, the selection pressure is increased by different methods, allowing to obtain the strongest target binders. Concerning the therapeutic approach, we performed 3 rounds of selection, and we determined the affinity of each round to the target and to the control, and we can observe that this affinity is higher than for the control. From the 3rd round of selection, we isolated 50 individual clones, and we evaluated their affinity. Here are the ratios between the affinity to the target and to the control, and we selected clones that have an affinity higher than the mean, namely 20 clones. The DNA of these clones was isolated and sequenced, and we observed that we had only 13 different sequences, and on these clones we determined their dissociation constants. In the future, we will select clones with a low constant, meaning a high affinity. Concerning the blood-brain barrier shuttling, the phage display was already performed and allowed the identification of 2 promising peptides. After to have evaluated their dissociation constant in the range of 10 to minus 11, we tested the competitive effect of natural ligands ApoB and ApoE on these constants. And we also evaluated the influence of pH and calcium on the binding. We observed that these clones are not destabilized by the competitors, but mostly for the clone 47, and that the decreased pH and the absence of calcium destabilize the binding from the target. These 2 clones were the most promising, and thus we decided to synthesize them. These peptides were tested on 2 cell types: human umbilical vein endothelial cells and hepatocytes. So peptide 1 corresponding to the clone 41 and peptide 2 to 47. We performed a semi-quantitative analysis of the fluorescence, and we observed that on one hand, the endocytosis of peptide 2 is higher than for peptide 1 in both cell types, and on the other hand, that for the peptide 1, its endocytosis is better in UVEC than in hepatocytes, but that's not the case for the peptide 2. Finally, we perform a colocalization of identified peptide with the LDR receptor on different areas of most brain slices. Peptides are coupled to rhodamine, and LDR receptor is highlighted by an antibody coupled to fluorescein. The merged photos show that our peptides have a good colocalization with the LDR receptor, meaning that they are specific to this receptor. To conclude, for the therapeutic strategy and targeting phospholipase A2 isoform, we obtained 13 promising clones, and a further selection will be done in order to identify the best candidates. For this purpose, we will test the ability of these clones to avoid the binding of phospholipase A2 to phospholipids. And for the best candidates, we will synthesize corresponding peptides and evaluate their therapeutic effect on cell models. Concerning the shuttling strategy and LDL receptor targeting, we already have 2 peptides synthesized, and they show an efficient endocytosis by UVEC and a good colocalization with the LDL receptor. The next step will be to evaluate their endocytosis and their transcytosis on cerebral microvascular endothelial cells and confirm the facilitation of access to the central nervous system. The final goal is to combine these 2 peptides in order to improve the access to the central nervous system for our therapeutic peptide and thus enhance the therapeutic effect in vivo. Thank you.
Okay. Other questions?
Thank you for your talk. I have just a pure technical question.
Yeah.
I just wonder, when you were talking about these calcium absent conditions, how did you reach this absence of calcium?
I'm sorry.
What do you mean by this? Calcium absent. How did you reach this absence of calcium? This technically. Calcium, yeah.
Because
Technically, how did you use some in big concentrations of
the
calcium
influence of
chelators,
calcium
probably.
on the binding.
Like BAPTA or EGTA or something,
but
some calcium chelators.
We tested the influence of calcium because we know that LDL receptor need the calcium, and so because we know that when we perform the phage display in incubation, in the incubation we have calcium, and then we tested the same but without calcium. So
yeah, this is my question. What do you mean without calcium? Calcium is very difficult to get rid of calcium completely.
Oh yeah, no, it's
So it's still some contaminating calcium is present there. So
yeah, but it's just to know, to have an idea to it's just to have an idea where clones and where peptide bind the LDL receptor. Because if it's the same, because we know that LDL bind the LDL receptor in presence of calcium, if peptide doesn't bind the LDL receptor without calcium, probably it is the same place on the receptor. It's just why we tested this. Okay.
Are there other questions? Okay, thank you.